Welcome, everybody. We're really floored by the attention. Just to let you know, we have about 700 people total and about 200 in person. So for us, it is the biggest program we've ever had. So thank you for coming and thank you for showing the interest. I just wanted to give you a little bit of an idea how this all started. So we are program managers and we are hired by ARPA-H to bring forth our idea. And as we join the agency, obviously we are driven, we have our first program, we know exactly as what we want. But as we advance, for me, this happens to be the fourth program, I still have ideas, but I fully acknowledge that they might not be the most impactful one. And we are a fully democratic agency. I was brainstorming ideas with my team back in September, and I was like, well, what should we do next? What's the most impactful program that we can think of? And one of my CETA, Scientific Engineering and Technical Advisor, Dan Bullock, the one in the red jacket, he goes, how about microplastics? And I go, well, how about microplastics? I am a physicist by training. I spent my career doing imaging physics. And of course, I pay attention to the news, maybe not so much. I'm not a health nut. I don't eat everything organic, but I started reading. And the good thing about this field, or the bad thing about this field, is that within a month, you read everything. And I'm very grateful for Matt Campen, who put together that wonderful little meeting, well, it wasn't that little, in Santa Fe in January, where you get a very clear idea of where the field is, what are the unmet needs, what are the most impactful things that we can do. And that's how this program came together. I'm very grateful to our agency director, Dr. Alicia Jackson. She will be here shortly. And she was nothing but supportive, right? I mentioned this to her back in December. It's like, well, this is the idea we have. And she looked at me, it's like, are you sure? And I go, yeah, I'm pretty sure. I read the literature. And from that point on, from January to probably March or so, like, Shannon helped. Again, I'm a physicist. I'm not a biologist. I don't do biology. And this program is really heavy in biology. And as a program manager, you have to be competent to oversee the work. And that really isn't me. That's Shannon. And you will hear from her as well. But I need you to understand that this program isn't just us. We are fortunate and lucky enough to be standing here in front of you. But the fact that this program exists is courtesy of our big teams. And I want to acknowledge my team, and then Shannon will get to introduce hers. So first, Dan Bullock. He's one of my CETAs. Adrian Ruskin, he's my lead CETA. And I will have to give him a lot of credit because the ISO is at least half written by him. And I have Mark Collage. He's the chemist on our team. So you will interact with him a lot. And Gina Perry, she is the contracting officer for this program on my side. And again, as you will see throughout the rest of the program, I will deal mostly with TA1. And Shannon, I will let her introduce herself and her team. She'll mostly be responsible for TA2 and probably most of TA3 as well.
You kind of forgot to introduce yourself. Alana Hanku, everybody. Good morning. I want to echo everything that Alana said. Thank you all so much for being here. The response to this program call has truly been immense. I've worked in the ARPA space for a long time. I was at DARPA for 10 years before coming over to ARPA-H about a year ago. I've never hosted a Proposers Day that has gotten this much attention. So thank you to everyone that is here in the audience today. And for everyone that's online on the webinar, we wish you guys could be here in person. But please don't be shy about submitting all of your questions to us, and we'll try to get them all answered. So again, I'm Shannon Green. I am a microbiologist by training, and I want to acknowledge the immense contributions of my technical team towards bringing this program to fruition. So I have my technical CETAs on the table to my left. We have Julia, we have Brenda, Mairead, and then to my front, and Ha-Jung. Brenda and Ha-Jung are actually very new members of my team, just picked them up last week. Julia and Mairead I've been working with for the last year. And this really has been a true labor of love. I also want to acknowledge Sean Rich. She's trying to escape the room. Sean is my business financial manager. And so for any of you guys that end up working with ARPA-H, she handles all the program logistics and all of the money. So she would be your very best friend. I also want to acknowledge my agreements officer, Miss Danielle Green. She's sitting here in the front, and 2 members of her staff. They work all the contracting wizardry. So let's go ahead and actually go to the next slide. And I want to talk about some agenda and logistics. Oh, am I supposed to change slides? Oh, thank you, Ryan. All righty. So as a quick reminder, actually, to everybody, this meeting is being recorded. So please silence your cell phones or set them to vibrate. It's really important. So after some opening remarks from ARPA-H leadership, we're going to dive into the meat and potatoes of today's event. Just going to be an overview of STOMP's vision, timeline, metrics, also how to work with ARPA-H, and putting together those proposal budgets. After we have that really monster morning session, we're going to take a break. Then we're going to reconvene for a Q&A panel. Right now, it's actually only scheduled for about 30 minutes. We can go longer if we need to, and everything will kind of flow downstream from there. We're going to have floating microphones that the CETAs will help to distribute, so please do not be shy about your questions. I have already been really impressed with the questions that have already come into the FAQ portal on the website. Please keep those coming. If you haven't been tracking that, that page gets updated about 2 or 3 times a week. If you haven't seen exactly how your question was asked when it came in through the portal, that's because we try to make the question, like very often a lot of people have asked a similar question, and so we try to merge the wording to make it accessible for everybody. Again, really want to encourage you all to make use of that Q&A panel today, because if you have a question, there's a 95% chance that someone else has that question. So please don't be shy. Yeah, I think that's about it for the morning. Then we're going to break for lunch. And then, as Renee mentioned, we have 2 exciting events for those of you who are here in person. And my sincere apologies for those of you guys who are virtual. We're going to have a TA3 brainstorming session. We're going to talk about this at length later on in the day. I'm going to foot stomp this, literally stomp. We are not soliciting for TA3 removals strategies right now. However, we think the time is right to at least start brainstorming on some of those ideas. And so, as Renee mentioned, when you checked in, you were able to get a different color-coded sticker for potential areas of TA3 removal strategies you might want to talk about. So after lunch, we are going to break out into those groups. We have a massive group here, so we'll probably end up splitting the room kind of into thirds for each one of the different areas. And then we can move the chairs around to sort of smaller groups of maybe 10 to 20 people, so we can actually have little conversations. Alana and myself are going to be floating around. Our CETAs are going to help also to manage and facilitate discussion. Then we will have some closing remarks from Alana and myself, and then we're going to open it up to the poster session. So thank you to everyone that brought posters here today. If you haven't set yours up yet, please make sure to do that, during either the morning or the lunch break. Use that to talk to your fellow proposer community and figure out how you might want a team and who you can learn from. And so with that, I think we'll introduce our fearless leader, Dr. Alicia Jackson. She has come to our agency in October of last year and has really brought the most immense enthusiasm and energy that I could have imagined to this role. She has been a fearless supporter of the STOMP program from the very first minute that Alana brought it to her. Anytime that I'm wandering through the halls and I see her doors open, we'll poke in and we'll have a conversation, and her face just lights up whenever we talk about this topic. It's gotten a lot of attention across the government, and it's just really, really exciting time. And we're so thrilled that you guys are all here. So I'll turn it over to Dr. Jackson.
Thank you. I guess we can use the podium, or I can sit down. I'll just stand down here. That seems a lot easier. I always have remarks, but I rarely use them, as my team knows. First of all, I just want to say welcome to everyone here. This is thrilling to see all the different folks here with different solutions, scientists, companies, startups, nonprofits, other parts of government. This is what we're here for. And we're not going to be able to solve this problem without all of you in this room. This is the team that's going to do it, and of course, all the folks online as well. This is literally, I don't know if Shannon and Alana already told you, but this is the largest Proposers Day we've ever had. And I think that speaks volumes to not only the criticality of the problem, but also the deep interest in solving it from all of you. I really need to give the credit to Shannon and Alana. They have done an incredible job building this program with lots of rigor, a lot of very thoughtful structuring of how we're going to go tackle this problem. It'd be very easy for someone to say, well, microplastics are a problem, let's go solve it. Let's just remove them. Let's just start removing, because that is the knee-jerk reaction I think that everyone has. But I think that they really realize that some of the major challenges that we have in this space are we need better measurement. If we don't understand particle count, where those particles are, what they're made up of, how they traffic in the body, what exactly are the biological harms that they may be causing, it's like boiling the ocean. We don't even know where to start. We don't know how much we're removing, where are we removing them from. And so I think the thoughtfulness that has gone in here, which is really developing gold standard measurement at the very beginning and throughout the program, is one of the most important things that we can do, not just measuring in blood, but in tissues and organs, which is going to be incredibly challenging and take a lot of thoughtfulness on the part of everyone who's come to this Proposers Day on how best to do this. And you're going to need to work together in teams. And we really want folks from other parts of academia, other fields, other parts of industry who've worked on measurement challenges before, but maybe have not turned their attention to this problem. And I know some of you in the room are very excited about working on this. I think the second part of this is really understanding how they traffic throughout the body. Understanding, yes, of course, we ingest them, food, water, air, but then how do they get into the other parts of the body and how much is really in those other parts of the body, and being able to consistently measure them across labs. That's going to be key as well. And then obviously removal, which I know lots of folks have lots of ideas about here, but I also think that's going to be a massive challenge. But I think it is all solvable. That's one of the great things about an ARPA, especially for those who haven't worked here before or worked with us before, is the ability to really go after very bold challenges in a very short period of time. And we can do that because of how we work with all of you. We're not a granting agency. We don't ask you to write proposals where you've already proven 80% of the work, and then we give you, 250, 500K and say, see you in a few years, let us know if you published a paper. That's not good enough here. We have a very big mission here and a large responsibility, which is to go after the hardest challenges, find the best people to go after them, and then be very focused, put significant resources towards them. But that means a huge amount of accountability on behalf of us to the American people, and also on account of you, to make sure that you're incredibly focused, to run as fast as you can, to bring the best minds to the problem. We can't do it unless we're able to do that. But it also means we're going to push you very hard. We want you completely focused on this. We want you to be coming to us, hopefully beating your milestones. So everything is a milestone-based contract. And for some people, that's going to be a new experience. And I'm going to tell you, it's going to feel a little uncomfortable at the beginning, but you will have the time of your life working with this team. I can promise you, and especially Alana and Shannon are incredible to work with. I know their teams love working with them, but it's going to be intense in the best way possible. And I can't imagine working any other way on this challenge. How could we not want to be intense? I don't think Shannon and Alana, or especially Shannon, let you know that we actually worked together before at DARPA for many years. And that was a pleasure to work together then, and we got to work on many things together and make many things happen. Alana, I met when I came to ARPA-H in October. There is no better place to work, period. Not just in the federal government, but anywhere. The ability to really go after the largest challenges facing Americans, facing our health, whether that be in biotech or healthcare, or other forms of biomedicine, and to be able to say, you know what, I think we can actually make a difference there. I think we can make a dent in the universe. And then to be able to go and find the best people in the world to work together to solve that problem, That is incredible. And luckily, some of us have been on teams that have been able to do that in a very short amount of time. And we're talking years. There is nothing that feels more fulfilling or more exciting than to be able to go after that. And look, I love my job. The PMs will tell you there is no better job than their job. And I think being a performer with ARPA-H, working on those very mission-driven programs, working very hard on those and very focused, is one of the most fun things you can ever do. I know some of you have worked with an ARPA before, and for those of you who haven't, buckle up. We'll do our best to work with you. I really want to encourage everyone in this room to think about joining. You have to be at the right point where you're ready for it, but maybe you've been feeling a little restless where you are. Maybe you have a big vision you've been dying to go after, and you can't because of, whether you're in an institution and you just don't have the latitude to be able to go after it, or you don't have the right people. Or I built 2 companies prior to this, and that was a great experience, but there's only so much you can do within a singular company because, you're focused on building the business. You can't always go down the other path you want to go down or do the deep science that you want to do. And this is the place you can actually do it and make something big happen and be very bold. It takes a lot of courage. It takes a lot of courage, it takes a lot of focus, it takes a lot of discipline to be able to go from vision to reality. And Alana and Shannon are really exemplars of that. So I just want to say, and in closing here, we're depending on you to bring us your best ideas, to go form teams with the best people. This is one of the reasons we have these proposer stays here and online so that you all can find each other. We're also going to hold you to a very high bar. You can imagine when we announce this, we get flooded with people saying, we've already solved the problem. If you have, that's great, but it's not enough to solve it partially. We're really going to stretch everyone here. It's not just what you can do individually, it's what you can do together. So we have a very high bar of rigor because we can't afford not to. We have to get this right. We have to get it right in order to solve the problem. And so we're really going to push you high. We're hoping that you want to stretch that far and run that far. We'll be right beside you. We'll make sure you have the resources that you need. We'll match you up with whoever you need to be matched with. We are on the team with you. When you become part of a performer team, we're in it with you. We want you to succeed. We'll do everything in our power for you to succeed, working together as a team. And so I'm so thrilled that we're kicking this off. One last story in closing, just so you understand how we work. Alana first talked to me about this program only several months ago. I really want that to sink in. Only several months ago. I think within like a month or 2, she and Shannon came and pitched it to us. It's kind of like Star Chamber at ARPA-H to get your program approved. It's no joke. We got it approved. I actually think the longest part of that entire process was finding a time for Administrator Zeldin and Secretary Kennedy to get together to do a press release. This is the speed we move at. I mean, that's just very unique anywhere, especially in the federal government, but even within a company, to be able to go straight from idea to launch to today is just phenomenal. I mean, that is a speed at which I don't know many others can actually move because we don't do things by committee. That's the other important thing here. We don't do things by committee. In order to be bold and to go after new ideas, you can't do this by committee after committee, working group after working group. It really takes people taking responsibility, having that rigor, Alana and Shannon, to be able to make those decisions. And so don't worry about going against dogma. That's the one thing I want to say here. We don't want to move slow. We don't want to do what everyone else has always done, trod those same paths or be iterative. We really need you to reach further. So thank you all again for being here. It's such a pleasure. This is my favorite part of the entire job, is being able to go and interact with all of you and hear your ideas and see your excitement and really bring us all together to solve a massive problem. Thank you so much. And I guess on to Shannon and Alana again.
Alrighty. So it's kind of a tough act to follow, and unfortunately, I'm going to make Andy Kiliansky do that. So he is the acting deputy director of the Health Science Futures Mission Office. Say that 3 times fast. Basically, Andy is deputy boss for the office that Alana and I work for. And he's joining us virtually today, and he's going to give you guys a little bit more of a flavor of the special sauce that makes ARPA-H, and also what makes HSF, in my maybe humble opinion, the best mission office at ARPA-H. So over to Andy.
Great. Thank you, Shannon. Thank you, Alicia, for kicking everything off. You want to hear from Shannon and Alana today, so I'm going to be very brief and go through just a few slides that frames our office in Health Science Futures, where we fit into the agency, and then some of our broader goals, beyond STOMP. If we can go to the next slide, one of the things that I've heard from proposers in my programs really across the agency is that even when people submit a proposal to the agency and don't get selected, going through that process of learning about ARPA-H, we're a relatively new agency, learning about that ARPA model, and really bringing teams of interdisciplinary scientists and experts together, that increases exponentially the chance of future success in proposing to ARPA-H programs or to DARPA programs or any of our sister agencies. And so, I really appreciate everyone being here today, and I'm really sorry that I couldn't be there in person. So the ARPA model, I think we already covered a lot of this, right? But we take a lot of our business practices from the Defense Advanced Research Projects Agency, or DARPA. DARPA was formed after Sputnik during the Cold War, when the Soviet Union strategically surprised our national security leaders. And we made the decision back then that we were not going to be strategically surprised again. And in fact, the United States, was going to be the ones doing the strategic surprising. Right. So this sort of technology supremacy on the battlefield has led to innovations that impact all of us every day, from the ARPANET, which ended up becoming the internet, to stealth technology and materials science, to autonomous vehicles. All of these things, right, impact our lives every day. And so for ARPA-H, really, I think our Sputnik moment is seeing the health disparities that we have here in the United States and globally, and really these challenges around really optimizing for health and reversing a lot of the long-term damage that we've encountered from our environment. And so STOMP really is that moonshot effort to do that, and embodies this ARPA spirit, right, of all of the programs that we launch here at the agency. Next slide, please. So, again, I'm not going to go into this in a lot of detail, but really, you heard from Alicia. She is there to approve programs and on a relatively regular basis evaluate how well they're doing. Beyond that, right, Shannon and Alana are your leaders, right? We work in a very bottom-up fashion at ARPA-H. So really, after a program gets approved, they have a lot of runway and leeway to go and execute that program, design it how they see fit, and select the best teams to work with at the end of the day. One of the other features of our agency is all of our leadership and program managers are all term limited employees. So Shannon and Alana have a sense of urgency. They're not going to be here forever at ARPA-H, right? And so they really want to make the most impact in their brief time here with the agency. And so that means things move fast. Fortunately, one of the reasons why I couldn't be there today, right, is because we want to get these programs started. We see these health challenges out every day, and we want to make sure that we're executing research to solve those as soon as possible. Next slide, please. So another really critical feature of ARPA-H is that, our customers are everyone. At DARPA, right, really, if they're working on a technology and it looks promising, they can get additional funds from the Army, the Navy, the Air Force to continue development of that technology into a capability, right, that the military can use. For us at ARPA-H, most of our customers are in the private sector, right? Whether those are patients, hospital systems, right, or companies who would look to take a technology that we prove a concept around here at ARPA-H and translate that into a product that patients can use. So it's really important as you're thinking about STOMP and what it means to really execute the goals of the program, to think also what you're building and the lifespan of that beyond really the short-term ARPA-H funding that you might receive, right? So that means, like, we at the agency work very closely with regulators like the FDA and EPA, very closely with payers and payees who are going to be reimbursing or using these services, and also industry, right? Companies that are going to be making products out of the innovations and discoveries you all make. So, Alana and Shannon will be bringing all of these groups together throughout STOMP, right, to really think about that long-term potential of the technologies we're developing here within the program. Next slide, please. So you're going to hear timelines later today from the STOMP team, but really we're at the start of the process, right? So Proposers Day is really when you all get together, form teams, learn about the program, meet the program team, and build for success, right, in the long term. There's a lot that happens after that, and I'm going to let the team describe that process. But really, you all being here today is incredibly important as you learn about really the moonshot goals that Alana and Shannon have laid out for you all. Next slide, please. So another feature of ARPA-H programs is that they are very actively managed, right? So Alana and Shannon are going to be meeting with you on a very regular basis, are going to be actively engaging with the science as it's evolving, and will be there to solve problems, right? But also be there to evaluate when approaches are not working. So our agency works its best when we fail fast, and we recognize that, right? And the resources that we might have pre-positioned against a technology or a approach, being able to downscope and really off-ramp from those approaches and bring them onto things that are demonstrated as being successful. So that active management process is something that's important for everyone to be thinking about as they're putting their proposal together and as they're building their teams. Next slide, please. So HSF, Health Science Futures, is one of 4 mission offices at ARPA-H. We all have our own goals. We all have our own program managers. We work very closely together, but right, the goals of the different offices vary. And so, it's great that you all are here for Health Science Futures, right? Really expanding what's technically possible when it comes to microplastics. But there's 3 other offices in the agency, and they are launching programs. They're having proposers days. And so we just wanted to call out, right, that by being here and meeting the teams at the Proposers Day today, you're really opening up access to the rest of the agency. And we really hope that you review and really follow the new programs that are coming out from each of these mission offices. Next slide, please. Okay, so you've heard enough from me. Really just to transition back to Alana and Shannon, right? Our goal in Health Science Futures is to create the future of health across innovation domains that remove limitations and really accelerate progress to products that patients can use. You can see from our program portfolio, it's incredibly diverse. We're in many different areas of science, technology, and health. STOMP is a really unique program, as Alicia mentioned, because it really combines helping the field identify the problem and quantify the problem, right, in TA1 here in STOMP, and then, right, launch into ways to diagnose, treat, and really create an environment that allows for industry and academia to solve these problems and build products way after STOMP ends, right, that are going to impact Americans' health in the long term. And so really appreciate everyone being here today. Thank you. And I will turn it back over to Alana and Shannon.
Thank you, Andy. Alana, you want to come back up? All righty. Next slide. We're going to launch into what you guys are all here to hear about, and that is the STOMP program. Alana, take it away.
Okay. Wonderful. I can even move slides. So welcome again. Here are the meat and potatoes, and I think we're a little bit over, so I am going to try to move a little faster. What if we could remove toxic microplastics from the body? That's where we started, but in order to get there, we have to solve a few problems along the way. So number 1, I don't have to tell you this. All of you in this room know we're producing a lot of plastics each year, and some of them stick around, and then they fragment and they end up in our water as well as in our body. There's all kinds of microplastics up there, polyethylene, PVC, polypropylene. We don't know yet. We know that as a class, they're questionable, but we have no good idea of which ones may actually be more damaging to the body. They enter the body mostly through ingestion and inhalation. They do pose a potential threat to human health. And again, I am a little bit of an outsider to this field. I am a physicist. When I started reading the literature, I had to scratch my head a little bit. So you read the literature that refers to human data and you see correlation, right? You look in disease and you see more microplastics in the plaques. You see more microplastics in diseased brain than in normal brain. You see more microplastics in cancers than in normal tissue. But this is correlation, and correlation doesn't mean causation, right? There may be a higher level process that causes dysregulation of an upstream process that actually causes both cancer and microplastics trafficking, right? Correlation does not mean causation. For example, in the summer, there are more people drowning. At the same time, there is more ice cream being eaten. It doesn't mean that eating ice cream will cause drowning. They just happen at the same time. However, if you start reading the animal experiments, then it becomes a little more dire. And in those cases, you actually learn that ingestion or inhalation of microplastics actually causes disease. So you have a control group, and then the animals who ingest or inhale microplastics, you see the plaques, you see the symptoms of neurodegeneration. Right, so this is where the field is. We have correlation at the human level, and we have causation in the animal experiments. And we see microplastics basically everywhere in the body that we look. Could we eliminate them? And you look in the literature, yes, there are some suggested methods in which you can limit your intake, right? You can avoid them. And I just retooled my kitchen after the Santa Fe conference, just spent $500 on that. But no matter how much I try, right? So I can try not to put hot coffee in paper cups, right? I can avoid doing that. But the water that we get comes through pipes, and the pipes are plastic, right? So there is only that much that you can do. In terms of mitigation methods, the only one that's potentially plausible is apheresis. However, again, because of the issues we have with measurement, it's not entirely clear to me that it does something useful, number one. Number two, it's actually not a very scalable method. Depending on where you go, it costs at least $6,000 per person. We cannot scale that up to look at all the Americans, for example. So where are we right now? Well, we're really in the dark ages, and it's a very interesting field in that many people have spent close to 20 years in this area. However, it's mostly from the environmental concern point of view, right? So you see toxicologists, you look environmental scientists, but it's mostly from the water point of view, from the marine animals, right? It's only maybe in the last 5 to 10 years that people have started to pay attention to what happens to us because we ingest this, and some of them tend to stick around. We have no idea what's the chemical composition that's going to cause more harm. We don't know how much, what's the threshold beyond which there is harm. We sort of learn that smaller is worse. So what we see over 1 micron usually goes in, goes out. What is below 1 micron tends to stick around. We also don't have a very clear idea of how the microplastics internalize, how they get trafficked. They get in, we have no idea, do they even get out? Honestly, I don't think we know as a field. So that's sort of the high-level view of the program. Our high-level view is we would like to limit intake and remove them, right? But you can't really do that given where we are, because number one, we can't really measure them well. So even if you claim you remove them, how the heck do you prove that you remove them? Because we can't measure them. So anybody can claim that they remove them, but not clear that they do. Number two is we really don't understand what exactly do we remove and from where, right? So to get to the end goal, we sort of have to solve these problems, and that's why the program is structured the way it is. So technical area 1 is measurement, and it spans all 5 years. And I will go in a little more details about that. So number one, we need a gold standard measurement. And as a field, it's pretty sad. We don't even have the gold standard. Most usually for everything we do, there is a gold standard, and then we need a cheaper, faster, better approximation of the gold standard. But in this particular case, we really don't have the gold standard. We have to develop that in terms of measurement, and then we have to develop an approximation of the gold standard, understanding that that may not be perfect, but it has to be fast and cheap. Technical area 2, which is the biological understanding of trafficking, will only run for the first phase of the program, which will be 30 months, right? So Shannon will go into more details of what we're requesting for that. The goal is to, basically understand what's the damage, right? We don't know. And at the end of that, we may learn a whole class of things. We can learn, number one, some of the microplastics. I'm calling them microplastics, but really they're nanoplastics. But again, just for simplicity, let's call them microplastics. We may learn that some of them really are bystander. They're not dangerous. Maybe they don't accumulate as much. They don't create health damage. There may be some of them that may actually be really bad. And in that case, we may, it's not us because we don't legislate anything, but the evidence that comes up during this program may actually be used to create legislation to limit those that may be toxic. The reality is that we cannot live without microplastics, and envisioning a world in which all the plastics are legislated out, that's just not going to happen, right? So there will be a place in between where things are not really, really bad and they're really not good. And we have to find the middle way on how to mitigate that middle road. And that's what technical area 3 is. I'm stating again, we're not opening technical area 3 because it's really not sane at this point in time. You can't remove whatever you cannot measure and whatever you don't understand. So here are the STOMP technical areas. I will deal mostly with TA1. I will PM technical area 1, and Shannon will take over and discuss TA2. So within the STOMP technical areas, there are 3 subcategories. Number one, we want the gold standard, right? We want the gold standard measurement that is not being questioned by the field. It's also the point to mention that this program will be run very differently than almost anything else we do, right? For everything else, everybody's a competitor. You go in your lab, you're not reporting anything because you're a competitor and you want to stay on the program and you don't want your competitor to not stay on the program. Not an option here, right? So this will be run as a circle of trust, meaning you all under contract will meet twice a year. You will meet with us. It's not only us. So we have CDC part of the program. We have FDA part of the program. We have NIHS part of the program. All of the government employees who have spent, many of them have spent careers. They will be with you in these meetings. We will have CDC basically perform continuous verification and validation. You're not going to be in your lab claiming you have discovered the wheel. We'll check the wheel every time, twice a year, right? We want to start from where the field is. We will start by gathering the scientific community at the beginning and establishing what is it that we have now? What is a reasonable dosing? What are reasonable lab measurements that you can take, such as you don't learn later that you're contaminated, right? So bear that in mind. We don't care if you have published or not. You will share with this entire group together with the government employees from many agencies and possible other people under NDAs. This will not be a secret program. Technical area 2 will be clinical measurement. So bear in mind, the gold standard needs to be fast. It needs to be, look at the milestones and metrics. It will be below 1 year. So most likely it will be something that already exists that has to be adjusted. You may have to adjust the protocols, what you dissolve, whatever it is. It has to be fast because without that, anything else cannot happen. And the ones who develop the gold standard, it depends how the teams balance out. you may have to do analysis for other teams because they don't have that super clean lab, and we don't want the results biased by their glass in their labs. So if you are the gold standard team, bear in mind that you may have to analyze a lot of samples for others, and we will establish that during negotiation. Under technical area 1.2 is, okay, now we have the gold standard, I need an approximation. I cannot spend 3 hours on the scattered stimulated Raman to analyze the sample. So what do we do to approximate the gold standard to a point at which it's good enough? At the end of the day, we want one test that's running under 15 minutes, under $50. And it's possible that it will be a scaled down version of the same technique. And it's quite possible that that clinical test would be something completely different. Right, so bear that in mind. We want solutions across the TA. So one proposal would actually mean responses to all three, right? So make friends fast. That's why you're here. Technical area 1.3 is conducting a study to evaluate if the microplastics that you see in blood or in body fluids that are very easy to access actually correlate to the levels that you find in tissue. In the end, we all fully understand that what we have in blood is really not the problem and what we pee out. The problem is what sticks. And honestly, we don't know right now if there is a correlation. If you look at all the studies out there, by the time something has gotten to the mitochondria, I'm not sure it goes out, right? It's TBD, and that needs to be answered. You have to think about how you design your study very carefully. It would be amazing it has to be a process that's feasible at scale, right? So we want this to be a solution for all of us. And if your study proposes brain biopsies, amazing. But let me tell you, you're not going to get a brain biopsy to see how many microplastics are in your brain, right? So whatever you propose for the study under technical area 1.3, where we look at the correlation between blood and tissue, it has to be something that's feasible at scale. We gave an example. You may consider colonoscopies because most of us, once they start getting gray hair, we get one of those every so many years, right? So it would be a relatively easy way to access tissue without being invasive. It doesn't have to be that. You can think about creative solutions, but it has to be simple enough that it can be deployed at scale. And the design of the study is up to you. Okay, TA2, I'm not talking. TA3, I'm not talking. Reference materials, very important. The field has spent most of its time dealing with polystyrene spheres, and there was a good reason for that. So 80% of the studies out there, the only thing you guys could buy were polystyrene spheres. Unfortunately, the field is now understanding that's not a good representation of our exposure. And as we started building up the program, we understood we have a problem, right? You can't continue doing the same thing over and over because it's not going to be a good recapitulation of our exposure. So we know right now that they're not all the same. We had to do quite a bit of legwork ahead of time, such as we will provide you with the relevant material. We will be closing the contract soon. We will put the entities under contract 6 months ahead of time, such as they have the runway to create the materials for you. So you will not have to worry about that. We will provide you probably 6 to 8 types of there will be 3 categories. So one under 200 nanometers, 1 200 to 1,000, and 1 to 5 microns, right? So assume you will have those in at least 6 categories. So there will be polyethylene, polypropylene, PVC, polystyrene, PMMA, nylon 6. Probably forgot some. Anyways, you have the most representative one, polyurethane. And the polyurethane comes because of the colon study. So you will go into the program and you will be shipped every so often the materials that you will need for your study. So don't bake that into our cost, into your cost. They will be on us. And we also, it is sometimes the studies don't get the same results, and some of the arguments is you use different microplastics. So that's one of the reasons we want to standardize everybody. There will be actually probably two, within one category, there may be two kinds, right? So produced by two people from different materials. So measurement, I'm probably over my time, so we'll take more questions. But again, number one is define the gold standard. We have pyrolysis GCMS. We have stimulated Raman. That's one thing. Either that make it better or propose something that realistically can become the gold standard within a year. We need that within a year. And we also need that clinical test that may take up to 5 years to develop. I will skip that. And these are the key milestones. Okay, look very closely at that. This is just a short synthesis of what we're asking for. This is not trivial. This is not where the field is. We're asking for the best-in-class method to have 10% variability, right? The field is not here. And you have to bear that in mind, how careful you need to be with how you handle samples, where you handle them, what's your processing to be able to get to that 10% variability. We're asking to quantify the mass, number of particles, and chemical composition for particles starting at 100 nanometers. We understand it may not be the same method. So at the minimum, we want chemical composition and number of particles, or we want mass and chemical composition, right? So there may be two tracks here, and you may choose one. The clinical quantification method, it's a little more lenient. So we're asking for detection of microplastics that are a little higher. We still want you to go for 100 nanometers. And again, it should be a fast method. Again, for technical area 1.3, we want at least 100 subjects enrolled per study. We want biological and geographical diversity, preferably people who have been exposed. So you expose more than others, potentially those who work in a chemical plant, such as you can infer some toxicity from that. And you will also have to validate, so you're going to have tissue and biological fluids and validate if there is any correlation. And with that, I'm done. Thank you.
All righty. Thanks, Lana. So we're going to switch gears a little bit and kind of talk about the biology. And I mentioned that my background is a microbial scientist, and I spent a lot of time at DARPA developing medical countermeasures to protect the warfighter from chemical and biological threats. And in all my time at DARPA, I would always think about product development from maybe starting with product design, maybe even in silico, then looking at, in a test tube, then maybe evaluating efficacy in cell culture, then maybe organoid systems, and then in animal models. In TA2, we're going to look at micro-nanoplastic health effects, starting actually inverting that pyramid and looking systemically first, and then kind of getting more granular down into the subcellular level. We want to be able to assess what's happening at the whole organism level, systemic changes, and then honing in on specific organ systems. Where are these micro-nanoplastics accumulating in the body? Where are specifically the different types of micro-nanoplastics, different sizes, compositions, right? How are they distributing to different organ systems? And then when we see where they go, assessing different functional, structural, molecular damage at the organ system level, and then diving super deep into that individual cell level, looking at the specific biological processes that are being disrupted. So cumulatively, TA2 proposers are going to need to address the who, what, when, where, why, and how of micro-nanoplastics. Just full stop. We're going to look at who. So what are the micro-nanoplastic polymer types, sizes, and morphologies that are most likely to bioaccumulate? What are the specific harms that they are causing, again, at these different levels of scale, whole body, organ systems, individual cells? When? Are there different types of or degrees of harm that occur through either acute or chronic exposures? The where. Where are they going? What are the organ systems? What are the cell types? Why? Why are they causing these specific types of harms? What are the cellular and organ processes that the nanoplastics are disrupting? And then finally, how? How are these micro-nanoplastics reaching their end targets in the body? And are there specific trafficking mechanisms that we might be able to potentially remove or reverse for removal? So I'm going to dive into metrics here because I think this is really important. And one thing that I actually want to foot stomp is please read the ISO. I even suggest, and I tell this to everyone that's proposing to a DARPA or an ARPA-H program, take the ISO and do a control find search and look for any instance where it says proposers must or proposers shall, and look for each one of those different areas because those are going to be really important parts that you're going to need to address in your proposal. And if you completely don't talk about it at all, it could even be seen as a non-conforming proposal. So please, please, please do this. We don't just write the ISOs just for the fun of it. We really do care about each of the different bullet points that are there. So TA 2.1 is looking first at that bioaccumulation and health impacts, right? We're wanting to look for proposals that are going to assess the system and the organ level metrics of harm, and also to look and define the half-lives of MNPs in the body. This is something that has not been established. Then in the last slide, I talked about the mechanistic framework, right? So we need to determine the cellular pathways that are impacted by MNPs and the mechanisms of toxicity. We need to be looking at what are the cell types and even the intracellular organelles that are affected. We want you to investigate and define those trafficking and accumulation mechanisms. How are the micro nanoplastics traveling throughout the body? Are those trafficking mechanisms different for polystyrene versus polypropylene? We don't know. It's a black box. And then try to identify if there are any innate mechanisms of MNP removal. There's some preliminary evidence that enzymes from the lysosome are capable of degrading some micro nanoplastics, but there's, to my knowledge, no evidence that that actually happens in vivo. So looking at some of the key metrics that are in the ISO, I just want to highlight a few. Solutions might leverage technologies that are developed in TA1 and advances in that gold standard technologies for imaging to be able to look at imaging and labeling of MNPs. This could include both stable as well as radioactive isotopes. We could look at different types of dyes, as well as multi-omic techniques to really understand the systemic and molecular harms that are being caused by the micro nanoplastics. We're also interested in looking at high throughput model systems, so different types of organoids, 3D cell culture systems, things like that, as well as computational techniques. I want to mention that across the board, these TA2 proposals need to include rationale for the in vitro and in vivo models that are proposed. Also need to be careful to define the methods for how you're going to down select those specific organ systems in TA 2.2. So in TA 2.1, again, looking very broadly at biodistribution, figuring out what types of microplastics are going where in the body, and then potentially assigning a risk matrix or a ranking of harms to figure out, okay, again, maybe there's certain polymer sizes and types that just don't seem to accumulate in the body. Okay, great, we can put them in a bucket over here where we don't care about them. If we do see micro nanoplastics accumulating in different organs, how are we going to prioritize which ones for subsequent investigation, really getting nitty gritty into the metrics and the details in TA 2.2. And then I want to also highlight, and again, Alana kind of hit a lot of these, but I want to make sure that this is very clear that across TA1 and TA2 solutions, there are some requirements that are shared, whether you're proposing to either of those TAs. All proposals need to discuss their methods for minimizing and mitigating micro nanoplastic contamination arising from standard laboratory practices and lab equipment. This is not something that we really need to worry about in a lot of biology, but like anyone that spent any time in the lab, we use plastic tips, we use plastic vials, we have nitrile gloves, our lab coats are made of frickin' polyester. So there are a lot of things we need to consider, especially when we're talking about measurement techniques, but also in the biological exposures. How are we mitigating and minimizing that microplastic contamination that might be coming from standard laboratory practices? Your proposals are going to need to address how you would do this. Proposals are going to need to make sure for any experimental methods and studies that you're justifying those methods and the study sizes. We are also looking for a candidate assessment, not only of the strengths of your approach. We know that you guys are going to come with your proposals and you're going to feel really passionate about what you have the best solution. But we also are looking for you to be candid about what are some of the risks inherent to your approaches and what are the mitigation strategies that you're going to take in order to address those risks across the program. We also want to talk about the exposure studies, and we want you to propose your best solution as to relevant doses and exposure methods. What we are going to do in the program, though, after negotiation and selection of performer teams, when we do kick off, we are going to invite our federal government colleagues from NIST and from FDA and from CDC and NIH to talk with everybody at kickoff about coming to a cohesive explanation or study plan that makes sense for assessing different types of exposures. Alana talked about this at length, so I'm not going to go into too much detail, but again, a lot of ARPA-H programs tend to be quite competitive, especially if there is a phase transition and there might be potential a down select from a number of performer teams. And so people want to be very cagey and not share their data. That's not how we're running STOMP. STOMP, again, is going to have a circle of trust, and we want everyone to be very open. And there's a lot of reasons for that. One, we see this as building a new field. This area is pretty small and kind of niche, and we really think that to solve these key problems in measurement and biological understanding, before we even then think about getting to removal, we need to bring people into the fold and get people thinking about micro and nanoplastics that maybe have never studied this before. And so we want this to be a very collaborative field building type of a program. So again, we're going to hold those semi-annual PI review meetings. We're going to learn from one another as we build this new field of science. And then finally, proposers, if you come to the table with your solutions, be aware that then you are committing, to potentially working with different partners across the STOMP program. As you noted, each one of the different technical areas has different subparts, and it's possible that only one of your subparts might be selected for funding. Again, I've been in the ARPA sphere for over a decade, and I think I can still, in those 11 years, count on one hand the number of times that I have fully funded a proposal completely as written. That is very, very rare. We do a lot of negotiations. We do a lot of trimming on our end. You'll do a lot of creative contracting work with our agreements officers. So it could be that, when you submit a full proposal, again, maybe only a piece of that is selected for funding, and we might have an overall vision of how that one piece could really complement something else. So when you come to the table, be prepared, to work with other members of the STOMP community. So again, we've just kind of talked about this a lot. We are not actively soliciting for TA3 proposals right now. We talked about this at length as a team, and we really came to the conclusion that it wasn't fair to the proposer community to have you guys submit your ideas for micro nanoplastic removal. Again, if we didn't have those gold standard measurement techniques to actually prove that we've removed what we said we were going to remove, and if we don't have that risk hierarchy, right, of different polymer types and sizes that we know are the ones we most want to target for removal. And also if we don't have a good understanding of where we need to remove them from, because all of those pieces of data are going to be really important in informing the creative solutions we're going to need to either remove existing ones from the body or prevent future accumulation. However, we're going to have a discussion section later on this afternoon to kind of talk about some potential areas. And when we were thinking creatively, among our team, we kind of came up initially with three sort of buckets that these removal strategies might fall into. And so one could be degradation. So thinking about creative solutions for degrading toxic microplastics into non-toxic byproducts that can be excreted out, or potentially thinking about if we're able in TA2 to identify some natural degradation processes that are already happening in the body, how can we potentially design solutions that accelerate those natural processes, right? One of the other things we talked about were potential strategies for expulsion. I'm really excited to hear what you guys are maybe thinking about in that area. We know the microplastics are able to get into the cells. Are there natural secretion processes or exocytosis pathways that we could potentially think about boosting in order to get them back out? And then prevention, right? Treatment is only kind of half the battle. We want to think about solutions that could maybe prevent micro nanoplastics from leaching into the tissues once they've been ingested. So we'll hopefully talk about that today. Again, when you checked in, hopefully you guys all got your stickers. If you didn't, make sure you go get those from Renee during the breaks. And then we'll regroup for that session and hopefully have a really good discussion. So let's talk about logistics. This program was launched with ISO released on April 2nd. The community really has already been fantastic about submitting questions to the FAQ portal. Again, I hope that you guys have been keeping tabs on that FAQ page. We do try to update that a couple of times a week. And so again, if someone has, you might want to also check that, because chances are if you have a question, someone else might have already asked it, and the answer might already be posted to that webpage. So please kind of keep tabs on that. So we're here today, April 22nd, for TA1 and TA2 Proposers Day. Solution summaries are due in just a couple of weeks on May 6th. So hopefully you guys already have some ideas of how you've already read the ISO. You might already have ideas of how you're going to propose solutions to TA1 and TA2, but this is your opportunity to meet fellow proposers, potential collaborators, and really kind of flesh out those ideas and submit those solution summaries, or our fancy word for abstracts, because we like to be fancy. Again, biggest piece of advice that I can give you guys all today is please read the ISO. Again, do that control find function, search for proposals or proposals must and shall. Please make sure you're looking for all those things. Again, also if you have questions about the ISO, something isn't clear, please send your questions to the STOMP inbox or to the FAQ page. Please don't send your questions directly to Alana or myself, because what we are going to do is just tell you and redirect you to go back to the FAQ page and submit it there. So just save everyone a step and just submit your questions directly there. Once the solution summaries come in, our teams are going to review those, and then you guys will get feedback as to whether full proposals are either encouraged or discouraged. Based on the number of participants that we have here in the room as well as virtual, I think we're going to have a lot of really excellent solutions that come in. And unfortunately, we're just not going to be able to encourage everybody. So the decision step, either encourage or discourage, it's not binding, but a letter of discourage does mean that a full proposal submission is highly unlikely to be successful. Once you do receive your feedback, if you get one of those encouraged letters, full proposals then are due on June 22nd. There are no extensions for my dog ate my homework. So please familiarize yourselves with the submission portal. You'll get practice with that at the solution summary stage. It's the same web page that you go to for both of those. Make sure you have your account set up and you know how to log in, and you give yourself plenty of time to get all of your materials uploaded. If everyone's trying to do it in the last minute, the site can potentially have hiccups. So just give yourself plenty of time. As a reminder, again, we are not soliciting ideas for TA3 right now. We are going to release a TA3 solicitation about 24 months into the STOMP program. And I think that is all that I have for right now. So how are we doing on time, guys? We're pretty good? Good. So next up is to take a quick break. Oh, and I guess I do have one last slide. So this is just kind of for y'all's reference. We're in the government. We love using acronyms. So please use this for reference. These slides are going to be posted onto the STOMP webpage. So if you guys, I know you guys have been taking notes frantically, this will all be posted online. So breathe. But this will kind of help you navigate some of the unique acronyms and terminology that we use. So I think actually we can charge ahead, right? And we don't need to take a break. I think we can just kind of go ahead into contracting. Okay, so we've talked a lot about STOMP, about ARPA-H, and actually kind of show of hands, who has worked with either ARPA-H or a DARPA before? Okay, so I see about a third of you guys, so that's awesome. So you guys have a little bit of knowledge of how to work with us, but there are a lot of people that did not raise hands. And so I want to bring up Danielle Green. She's one of our agreements officers. She is going to give you guys some clues as to do's and don'ts when submitting proposals to ARPA-H, how our proposals are reviewed, what's the contracting process, what does all that look like, and how to work with us. So Danielle, come on up to the podium, please.
Okay. Hi, everyone. As Shannon introduced me, I'm Danielle Green. I'm an agreements officer with ARPA-H's Business Innovation Division, and I support Shannon. So I'm just going to be giving you a quick kind of briefing and outline of the STOMP Innovative Solutions opening. For short, we call it an ISO or an ISO. So just to start with some ISO basics, ISOs are designed to enable merit-based awards, allowing the government to focus on the technical quality of the submission rather than the price. This means that we're looking for the best ideas, not necessarily the lowest cost. Rather than prescribing a single solution, ISOs encourage a wide variety of approaches to solving problems, and they offer flexibility in the types of awards that can be considered. So some highlights of the STOMP ISO is that all of the awards will be made as other transaction agreements. And all types of entities are encouraged to participate. There are no funding limits for individual projects. So you'll want to ensure that whatever solution that you propose, the cost associated with that solution is comparable to what that work is. We do strongly encourage questions, and all answers to questions, whether they're your direct question or kind of a consolidated of similar questions, they will be posted publicly. As Shannon said, they are posted on our FAQ page. Shannon already kind of touched on this, but solution summaries are mandatory. There are two technical areas that the ISO calls out. It's technical area 1 and technical area 2. So if you're submitting a solution summary, you can submit separate solution summaries for TA1 and or TA2. However, at the full proposal stage, you may only submit one full proposal for either technical area as the prime. Proposing entities may also be a part of multiple teams as subcontractors. And then this was kind of already touched about collaboration, but in order to meet the program's goals, if needed, ARPA-H may want to establish associate performer agreements, APAs for short. And essentially they're just formal agreements that set terms for how performers will collaborate with one another, whether that's sharing necessary data or resources, and they help teams coordinate, share information, and also avoid duplicating efforts across related projects. Please keep in mind that a submission, whether that's a solution summary or a full proposal, if it doesn't conform to the ISO requirements, it may be deemed non-conforming and it could be rejected without review. So as Shannon previously said, read the ISO carefully and ensure that you're conforming to all of the ISO requirements. And then, of course, if you have any questions, please reach out to the STOMP inbox or go ahead and email, just the STOMP inbox for now, and then also refer to the FAQ page. Okay, so we did say that we anticipate awarding multiple other transaction awards. So if you're not familiar with other transactions or OTs, we like them here at ARPA-H. They are flexible, collaborative, and legally binding agreements that are not traditional procurement contracts, they're not grants, and they're not cooperative agreements. They are modeled more closely on commercial contracting, and they're not subject to all the federal acquisition regulations, otherwise known as the FAR. This also allows for more negotiations and partnership between the government and the performers, and it makes it easier to just share resources and knowledge. So if you haven't seen what an OT looks like, we do actually have a sample on our ARPA-H webpage. You can also scan the QR code here for direct access to that sample. Okay, so if you look at ISO Section 3, this outlines the eligibility requirements. So if you're wondering who can actually apply to this ISO, it's all responsible sources that can meet the government's needs. So that can be nonprofits, academia, commercial organizations, or small businesses. We do have in this space federally funded research and development centers, also known as FFRDCs. There's also other government entities, and they are not eligible to participate as the prime or a subcontractor under this ISO. However, if you are proposing to the ISO and you have an FFRDC in mind that has a unique capability that you think your solution is unachievable without it, you can submit as part of your solution summary documentation, demonstrating its necessity and that all other options have been exhausted. And then ARPA-H will go ahead and review that documentation and then determine if the inclusion of an FFRDC or another government entity is absolutely necessary. If you are providing contracted support to ARPA-H already, you are not eligible to submit to the ISO because that would be a conflict of interest. Okay, so regarding US-based and non-US-based entities, Priority is given to US-based entities, but non-US-based entities can also participate if they're compliant with United States law and regulations. However, there are certain foreign entities and individuals that are excluded, and they are listed here on the slides. And then finally, all proposals have to have an active SAM.gov registration and able to participate throughout this whole entire process. Okay, so if you look at ISO Section 4, this talks about the submission process. As previously stated by Shannon, a submission of a solution summary is mandatory. This has to be no more than 4 pages. It's submitted to the ARPA-H Solutions portal. And then you would just follow the guidance for that submission in ISO Appendix A. And then ISO Appendix B outlines the full proposal submissions, which are also submitted to the ARPA-H Solutions portal. Section 5 speaks to the review process. So how are these submissions reviewed? Solution summaries that are within the ISO scope are independently reviewed. And then based on that review, you'll either be encouraged or discouraged from submitting a proposal. I do want to say that regardless of the feedback, you are still permitted to submit a full proposal. But as Shannon said, it's highly unlikely that you may be considered for award negotiation if you're previously discouraged. Full proposals are also independently reviewed. However, these are evaluated against specific evaluation criteria that is outlined in the ISO. And it is noted that any proposal that's submitted is not compared to one another. They're independently reviewed on their own merits. Again, you will be notified each step of the process. All right, moving on to how we evaluate full proposals. So when it comes to the evaluation, it's shown in the ISO that there is 3 main criteria that evaluators will be reviewing your proposal against. And that's in descending order of importance. So first is the overall scientific and technical merit. Reviewers will look for evidence that your approach is innovative, feasible, and complete with clear deliverables and risk mitigation strategies. Second is your team's capabilities and/or related experience. So here, reviewers will look for evidence that your team has the skills and the background needed to accomplish the proposed work within the budget and the schedule, including clear descriptions of key personnel qualifications, demonstrated experience in similar efforts that have already been completed or are currently ongoing, as well as identification of other government entities that may be involved. And the third and final criteria is the assessment of the proposed cost price. And this just ensures that your cost for your effort is reasonable and aligned with your technical goals. Additionally, cost realism may be conducted to ensure that the costs are not only justified, but that they reflect a sufficient understanding of the cost and the effort needed to successfully accomplish the proposed technical approach. And then following the evaluation of your proposal, you will be notified whether or not your proposal was selected for award negotiation. So here's just some final guidance. Shannon kind of already touched on this as well, but to wrap it up, ensure that you keep monitoring SAM.gov for any ISO updates. Remember that only formal amendments to the ISO can change the actual terms of the ISO. So just continue to keep a lookout for that. Make sure that if you're not already registered in SAM.gov, that you register as soon as possible. Carefully read the ISO and make sure that your submission not only conforms to the ISO requirements, because non-conforming submissions may be rejected without review, but you want to also note the key deadlines, right? When is the solution summary due? When are the full proposals due? What is the deadline for the Q&A? Additionally, read the ISO and the FAQs before submitting questions. As also stated, a lot of the times we get repeat questions. So take a look at those documents first before asking your question. If there is any contradiction between the FAQs and the ISO, always defer to the ISO, but go ahead and let us know if you feel there's a contradiction, and we'll go ahead and investigate that and make sure that there isn't, or correct anything if we need to. All questions, they should be clear. Ensure that they don't contain proprietary information, and that they're sent to the STOMP inbox. that's kind of my quick run through of the ISO. I'm sure I don't know if all of you have read it, but again, go ahead and just carefully read that again. We're here if you have any questions. And are we going to introduce
Sean? Yeah.
Sean? Okay, yeah. So next speaker will be Sean, Shannon's business financial manager, and she's going to be talking about the cost and administrative requirements. Thank you.
Hi, good morning, everyone. My name is Sean Rich, and I'm Shannon's business financial manager. Before I dive in today, I wanted to note that STOMP is a unique program in that it has 2 program managers. So on that note, we also have 2 business financial managers, myself and Miss Jennifer Myers. Jen couldn't be here today, but she's equally involved in the program. And between the 2 of us, we've got you covered on the cost and administrative side. So I know this is when all the scientists in the room start to not pay attention, but please, there are a couple changes if you've worked with ARPA before that are unique to this program. So with that, let me walk you through the cost requirements for STOMP, and I'll keep this straightforward so you know exactly what to expect. Okay, we'll start with the solution summary stage. When you submit your solution summary, you'll need to include a basis of estimate or a BOE. We've provided a template in Appendix A, so please use it. A couple of important things to keep in mind here. First, the BOE, it does not count towards your 4-page limit, so don't worry about it eating into your narrative space. Second, we need a single consolidated BOE for your entire team. We don't need separate budgets for each team member, 1 document per team. Also, please be aware that the HHS salary cap applies and that you'll need to break out your total cost by technical area. We've also given you a table for that. So I want to set expectations here. The solution summary stage is the high-level cost overview. We're not looking for a fully substantiated budget. We know it may change when the full proposal comes in. That'll come later. Okay, when you move into the full proposal stage, the cost requirements become a little more detailed. Volume 2 of your proposal requires 2 documents, a cost proposal spreadsheet and a cost proposal narrative. For this spreadsheet, you'll use the ARPA-H abbreviated cost proposal spreadsheet template, which you'll find in the ISO Volume 2. I want to highlight the word abbreviated here. This is unique. You're not required to provide backup documentation like quotes or vendor estimates for materials or equipment over $10,000 at the full proposal stage. We've done this intentionally to streamline the submission process. If you are selected for award negotiation, you will need to provide that supporting documentation. So just keep that in mind. Every team member that makes up your team should use the full template, and that your budget should account for all the phases of the program, each priced separately. The spreadsheet has multiple tabs. You'll see it covers personnel, materials, equipment, indirect costs, ODCs, consultants, and more. Okay, separately, for the cost proposal narrative, there's no template for this one. This is where you'll provide your value analysis, supporting information like your indirect rate agreements, labor justifications, fee or profit rationale, subcontractor consultant justifications, and any cost share contributions if applicable. Okay, so now some suggestions and reminders starting with our dues. Do provide all requested documentation and information at the cost proposal cover sheet. Do include a cost estimate of total funds requested from ARPA-H and any cost share. Break your cost down by task, fiscal year, and phase. Do itemize your subcontracts. Subcontractor cost proposals, again, need to be prepared at the same level of detail as the prime. It's the prime proposer's responsibility to compile all subcontractor proposals and show how these costs map to each phase and each task. If you're using consultants, include an agreement or the document that verifies the proposed loaded daily or hourly rate. And finally, do make sure your proposed costs are realistic, consistent with your statement of work, and reflect a clear understanding of the level of effort needed to execute your technical approach. And now for the don'ts. Please take these seriously because they'll save you and us a lot of time. Don't change the formulas in the spreadsheets unless it's absolutely necessary. If you must adjust them to match your business practices, please double check that the total amount tab still reflects your costs accurately. Don't delete any rows, columns, or worksheets. Even if you don't need them, just leave them blank or hide them. Deleting them breaks the formulas and then increases the processing time on our side. Don't submit any other format other than the Excel. And importantly, don't try to buy in. We recognize that overemphasizing cost can push proposers to a low risk idea, minimize uncertainty, and junior staffing just to look competitive. We discourage all that. We want your best technical approach, appropriately resourced. So that wraps up the cost and administrative requirements. If you have any questions again about any of this, please email the STOMP inbox. Thank you for your time.
Okay, so welcome again. We will take questions. The plan is to take questions for half an hour. But if we need to extend, we will extend. I think that is the best place to get your questions answered. Again, we're doing our best to field frequently asked questions, but it may take a while. So now's the time to ask everything that goes through your mind.
Hey, Kyle Dowdrick, University of Notre Dame. This is a question for TA 1.3. I think it's great that we're using these standard micro nanoplastics, but I wonder, is there something that should be in place to have a standard collection approach for when we do the clinical piece to minimize background contamination of plastics?
That's a good question. And again, we are going to run this as a circle of trust. So at t equals 0, there will be a meeting between everybody who's selected and essentially everybody who has been in this field for a long time. And we will decide at that point, again, although I would like to claim I'm an expert, I'll have to acknowledge that I'm not. I have not spent my career in this field. I have read the literature though. So I suggest we all meet at that point and decide where do we start. And if it is decided at that point that there will be a standard way to collect samples, then we will follow. Again, the one thing we're enforcing is we want to certify everybody within a month that however they're doing their sample analysis is plastic free. So if you do not have an access to such facility, then the TA 1.1, the gold standard performers, when they sign the contract, they will agree to do the analysis for however many samples are needed, because that's the gold standard that we have, right? So that's in terms of analysis. The collection, again, because we're not enforcing how the clinical trial will look like, it may not be possible. If somebody wants colonoscopies or, uterine tissue, it may not be trivial to standardize.
One other thing that I would just kind of add to that question is, while we want to develop more standardized collection and microplastic mitigation techniques with our selected performers when we kick off the program, in your proposals, what that means is please propose to us how you would go about trying to minimize or mitigate micro nanoplastic contamination kind of at all stages of the research, whether that's TA1 or TA2. Now, your exact method and way of doing things, we might end up merging that with other groups if selected for funding when we kick off and we have this meeting where we all kind of come to Jesus and figure out what we're going to do across the program. But at the proposal stage, just propose to us your intent and how you want to approach that contamination issue. Does that make sense?
Hi, my name is Akuya Asawuku, University of Maryland, College Park. So I was hoping to get a little more insight on the synergies between TA2 and TA1. Is it possible to think about doing a TA2, but then later on doing more into the TA1, which is the gold standard, trying to figure out things later on, because the TA2 does occur in the first 30 months, right? So have you thought about this and the synergies about the different tracks and the timing of the different tracks as well?
Yeah, so we're definitely, synergy is kind of the name of the game in this program. We are soliciting for separate proposals to TA1 and TA2. However, if you remember from Danielle's presentation, at the solution summary stage, you can submit your solutions to both technical areas. There will be separate documents, so you'll have a separate TA1 solution summary and a separate TA2. You can be at the solution summary stage, you could theoretically be prime on both. However, at the full proposal stage, you can only be prime on one. So if encouraged to move forward for both TA1 and TA2 ideas, then you'd have to decide which one would your group want to be prime on, and then you'd maybe move to being a sub for the other. But yeah, we definitely encourage synergy. And so that can occur, if performers are on both TA1 and TA2. And also, even if you're, a group that's only doing TA2, you're going to still be probably collaborating with TA1 teams because they might be performing some of the measurement techniques, for your group.
Hi, Gustav Ganguly from University of Arkansas. And my question is specific for TA2, and I'm an inhalation toxicologist. We have seen while doing nanoparticle-based research, I'm guessing that might be very similar in this case, that size difference and difference in shapes would matter a lot. So what I'm wondering is that though you are going to provide 6 different kinds of micro nanoplastics, but is that the same material? Are you guys going to provide us with different shapes and sizes of the same thing?
The answer is yes. So more information would be provided, during the negotiation phase, because then we'll already have entities under contract that are going to be producing the reference materials. So there is going to be a variety not only of polymer types, but also size distribution. So as Alana mentioned, we're going to have 3 sort of size buckets, one centered around kind of 200 nanometers, another that's kind of between 500 nanometers to a micron, and then 1 to 5 microns is the goal. We also know that, and one thing that we learned when we were researching this program and figuring out what the structure was going to be, that you can't also just go to like Sigma Aldrich and like go to their catalog and buy like a specific size or specific shape of particles. And like the only thing that really you can buy are the polystyrene spheres. And we know that when particles break down in the environment, when plastic waste breaks down, it doesn't perfectly break down into spheres. And so those might not be super relevant. And so we are challenging the entities that would make the reference materials to make things in different shapes. So more irregular shapes, more shard-like particles that would be representative of what we know happens in the environment.
Got just a follow-up question. And would you ensure that this micro nanoplastics are endotoxin free?
Yes. The other thing that is in the ISO, again, encourage you all to read the ISO, that you are also encouraged to, in addition to the reference materials that we're going to supply, you can also propose other materials that you want to layer on top of that as well. If you have your own source or methods of generating those materials, that is also fine. But what we want to do is make sure that we're grounding the program in a common set of materials that everyone can use, so we have more comparability across the different performer teams and across the different methods. We want to try to standardize this field somewhat. But if you have other materials that you want to bring into play that you can make in your own labs, please do so.
Probably a last follow-up question to that, sorry for that, is that how are we going to maintain the consistency across labs when it comes to dosimetry, if you're doing an inhalation exposure or an intratracheal instillation? That was my last, thank you.
So again, that's the part of the magic meeting we'll all have at the beginning, at the kickoff of the program, where I expect will be 50 to 100 people. And we will all agree at that point at what have we learned from the literature, what's a reasonable dose for short-term, long-term exposure, how it should be done, and then everybody will agree to that. Because we don't want the results to be different just because people use a different way, right? So we want to baseline everything together.
Would add on to that in addition to standardizing, some of the dosing and exposure routes, if there's something that goes above and beyond that, that's like a unique, extra capability or an extra control group or something like that, then that can be phased in as well. But again, we want to try to ground this as much as possible. Next question.
So Vito Quaranta, Northeastern University in Boston, and I have a question for, I suppose, Shannon on TA 2.1. You're looking at organ systemic distribution. Is it possible for an applicant to focus on a particular organ, like the brain, to be clear, just to pick one? And then the other thing is, the work obviously must be done, especially mechanistically, must be done in small animals. Do you expect validation in humans, on human tissues?
So TA2 is not going to have a specific validation in human studies. It's really not ethical to expose humans to more microplastics. And also, we don't really have a control group of none of us that have been exposed. when we're talking about the biodistribution, we can certainly have solutions that encompass, focusing on specific organs. However, we'd want to also ground that, based on what we know from whole body distributions, right? And what specific polymer types and sizes actually go to that organ. So you kind of need to, the way I think about it, and I'm happy to be wrong and would entertain, proposals that are maybe a little bit different, but it makes the most logical sense to me to think about whole body distribution, and then starting to hone in across biological scale to sort of more narrow areas. And so if a group has a specific focus on one particular organ system, then I would highly encourage you guys to talk amongst yourselves and think about teaming and how you can have complementary expertise so that a proposal is not focused on just one organ system, because that wouldn't be conforming.
Hi, Evie Voltura from Texas A&M University. I had a question regarding TA2, mostly related to diets that we're feeding the small animals, because that could have different levels of contamination. So are you guys interested in groups that can formulate their own diets so that's consistent across the board? Because I see that being another source that could cause variation in results across teams.
Sure. So, I think that, we don't want like every single performer team to be doing the exact same thing. So if there is variation in diet, then that's going to give us more information, about the different health effects of micro nanoplastics and how that intersects with food. Now, specifically about contamination of plastics, in the food, then that could be something that we would want to address. Again, we're requiring that TA1 and TA2 proposals do discuss their mitigation strategies such that the micro nanoplastic exposures are coming from what we're intending to provide, to those studies, as opposed to what's extraneous.
And one more follow-up question. For the small animal models, are you only wanting mice, or are you agnostic?
It's up to you guys.
Okay, great.
Yep. Just need to justify your models, both in vitro and in vivo.
Just be mindful that we don't want to swamp the universe with more microplastics that we produce for you. So I would rather you don't feed pigs nanoplastics, because that will take a lot. So bear that. We know we're not forbidding anything but mice, but because of practical reasons, I would encourage you to think more of small size.
Hi, Justin Morrow from University of Cincinnati. There's a specific question on TA 1.3 study. So I think you're clear in the purpose of that study, and it's not going to overlap too much with TA2, different purposes, but your specific requirements for at least 100 subjects or a power analysis if we're going to do something different, does that change if we want to target, for example, one system versus smaller studies across multiple systems? How would you see the 100 subject total? Could that be distributed over others? Would you just expect a power analysis, or is each one multiplied by 100 minimum?
Well, again, so I had explained the reason for that, right? So right now we don't know if we can look at urine or blood and understand how exposed we are. So we needed something else, and that's why we asked for different organs. Now, if you think you're powered enough and you want to split your 100, that's fine, but make sure you make an argument as to why you think you're powered enough.
On each
on each subcategory, right?
Yep.
Yeah.
So we expect, again, at least one of the subcategories. So that choice gears towards TA3, right? So our fundamental question, we looked at each other and well, what if they're not correlated? Then what? And we said that
the TA 1.2, sorry, we're premising that TA 1.2, our fast and cheap diagnostic, would probably be out of blood or urine. Yeah. And then we were like, oh, shoot, what if they don't, right? Then what if we can't use blood or urine? And so then we were like, okay, what would we use if we can't use blood or urine as an approximation of what's going on in the tissue? Since that's where TA 1.3 comes in.
Yep.
Right, then at least one of the organs that you propose has to be something that is accessible, that foreseeably, right, in TA3, we're going to find out, well, some microplastics are not good to be around. And we may want to screen people and try to remove microplastics from that particular person. We know we're not all as loaded as each other, right? And that one type of tissue has to be accessible.
Yep. Okay,
Don't
thank you.
just do brains and, hearts, because
Right.
I will not have a heart biopsy for
Right.
that.
Yeah, exactly. Thank you.
This is Ting Gu from the University of Oklahoma. I have a TA2 and also specific for general question. So we've using human organs already, has liver in the lungs and the kidneys, and then expanded to heart to study the toxicity as microplastic. But question is the real world plastics from the environment is not virgin plastics. We know there's a plastics intrinsic, additives or absorbed. So the STOMP or follow-up other funding will be consider study the real world microplastics. Or if you're providing the microplastic standards for all of our study, were you considered to adding this for co-toxicity study?
Yeah, I mean, we can both kind of answer this a little bit. So again, because STOMP is going to be looking across different polymer types and at different sizes, we already kind of have a pretty complex matrix of what we're looking at. And so adding in the extra variety of looking at additives was going to kind of boil the ocean and sort of be too big. And so we're like, we have to start somewhere. So we set some parameters. Now, to your question about, weathering, that is something that we are wanting to have as part of the reference standards, materials that do have some sort of either natural or artificial weathering to kind of mimic some of those processes that you might approximate our actual natural exposures to. As to, follow-on funding, with ARPA-H, like we tend to kind of do one-shot programs where we set that moonshot goal and we, take a really high-risk investment. And then the hope is that other agencies within the federal government might be able to fill in some of those gaps that we're not able to tackle because we're doing a lot, but we can't do everything. And so I think that's be a really great question for, other government agencies to be able to kind of follow on and some of the work that we're leading here.
I see. Thanks. But I know it's a really complex system. Yes.
So let me follow up. And I think that would be a really interesting conversation to have. Again, we will be contracting and providing microplastics to you guys. We can start from standard materials, and the advantage to that is we know what we have, right? It won't be weathered to start with. We can ask them to be weathered through UV or whatnot, right? The advantage of that at least you know what you're studying, right? We can also start from beachcombed plastics, and that would be in a sense more relevant. The problem with that is that you cannot guarantee round-to-round repeatability. And then again, we get into a point at which we cannot compare results because you had a different batch. And we've been going back and forth among each other, and we likely will do the same with the entities we put out. It doesn't matter to us what we do, but I think it is important to be able to cross-reference the results. And at least that's why I'm thinking a good fraction that we will provide you will come from standard materials and will come with, obviously processing can introduce variability. We will give you all the characterization data that we will do. There will be 6 to 8 types of characterization. I'm not sure we're going towards ocean plastic collected off the beach because it has that inherent lack of repeatability problem.
Yeah, I'm more because we are interested more is in the like water, the cups we drink, then plastics, the boxes we put in the food. So everything's more relevant to human contact. These things I think is made more prompts to go into the human body. So whether we can study, I know it's a complex because you cannot control every addictive for the manufacturers, but some common things like where we starting from there and going towards. Or I agree with you, for more control the variables, we'll be able to compare across boards. Yes.
And again, I want to make sure we said this before, and I'll kind of say it again, that if there are materials that you really want to work with that are beyond what we are supplying, then you can add those in. You're still going to need to work with the materials that we give, but if you have other ideas or other sources, then that is a welcome addition.
Thank you.
Yeah, I'm Justus Ndekife from Vanderbilt University. So my question is, could you comment on what are your expectations for the commercialization requirements to make a very strong proposal?
This program is fairly early. Like a lot of ARPA-H programs, we span technical maturity. So there are some ARPA-H programs specifically coming out of like the Scalable Solutions Office that are looking at, how can we take technologies and actually scale them to like all Americans in the course of a program, right? And so commercialization is very, very heavily weighted in those programs. This program is much more to the left, much lower technology readiness level. However, we do want to think about, though, how these technologies, actually would scale both on the diagnostic side as well as on the therapeutic side. And so if you read the ISO, there is a section on commercialization requirements that talks about teams during the program assembling a translation advisory board that can help the team to think about, again, how they're going to move their technologies from the bench actually to the bedside. And so there's a table, I forget the page number that it's on, but there is a table in the ISO that talks about different commercialization requirements and what needs to be included in the proposal.
Yeah, again, and the most important part, I guess, is the clinical test. Like, I think it would be to a proposer's advantage if they showed that there is a clear avenue through which that particular diagnostic method can be commercialized. So that is important. Maybe less so on the gold standard, but more so on the clinical test side. And obviously on the therapeutics, with the understanding that most likely you won't have a therapeutic by the end of the program, right? But the diagnosis side, I think there is an expectation that whatever is developed will actually be commercially viable by the end of the program.
Thank you. My name is Loza Tadessa from MIT. My question is regarding the team. What do you look for in a leader in the team? How many teams do you anticipate to award in your program this cycle? And if startup companies can be a lead for a team?
I can start with answering the last question first. So yes, we are able to fund anyone, whether that's a large company, small startup, academic group, whether that's an academic that's a very large, well-established team, or like a new professor. We can work with anybody. That's the great beauty of being in a place like ARPA-H. However, because if you read the ISO, there are a lot of moving parts in both the TA1 and the TA2 proposals. And so it's likely that there will be some teaming involved. And so a team could mean one PI in their lab teaming or pairing up with another PI from the same institution. That makes things maybe logistically a little bit easier. However, we also see teams where there might be a company that partners with an academic, or maybe multiple academics at different institutions that all work together. And if that's the case, then we want to see not only do you have a strong PI that's able to drive the technical direction of that team, but also on the project management side, like who is going to be responsible for making sure, that we're managing the budget, like who's going to deal with coordinating, right, from different institutions, who is going to be the key for making sure that deliverables are, sent to the government. And so like the more complex that a team grows, the more that we do look for that project management expertise. And that doesn't necessarily need to be the PI, that can be two separate people, but we want to make sure that that's not neglected because these projects can be really big. The other thing, I think in press releases, you guys have seen the overall budget for this effort has been published, so it's publicly available. Remember, there are 3 technical areas, though, so take that dollar value that you've seen in press releases and maybe think about dividing that by 3 for each TA. But then remember, also, we're probably going to be funding multiple teams per technical area. So you guys can do a little bit of math and think about we're not going to give you exact dollar values for what we're looking for. Again, Danielle mentioned that we want you to be proposing the best science, full stop. That scientific and technical merit is like the main criteria that we look at. However, we do also look to make sure that the budget is matching the work that's proposed. We look to make sure that it's realistic. So, propose to us your best technical solution, tell us what you think it's going to cost, but also you can bear in mind and do a little bit of math and know that we're not going to be funding just one TA1 group and one TA2. It's going to be multiple teams.
Hi, my name is Amari Walker Franklin from RTI. So actually right along the lines of that question, for TA 1.1 is, the start of the gold standard method, but also potentially teaching other labs or doing additional samples for other labs for TA 1 or TA 2. Is there any anticipation of the number of samples that we could at least start our budgeting for estimations, or number of labs that we might have to train, just to give us a ballpark idea for how to start with costing?
I mean, it's really hard to do that because we don't know who will select, right? So in some cases under TA2, we may actually have people who are very competent, who already are associated with plastic-free labs. And in that case, you'll get none. Or you can get somebody who will send you all the samples. So, take 1,000 samples, I don't know. But that will be sorted in the negotiation process. Right. Nobody can give you a better answer. I wanted to follow up on the earlier question a little bit. So as an ARPA, we are actually, it doesn't matter to us if you are a full professor or an assistant professor. I had suffered through NIH pain, and I have been told directly, who do you think you are to write an R01 without having published five papers? And I stand against that, right? So propose your best science. However, we have also found the problem that as a junior faculty, if you're bringing the best stars, you can't rein them in. And they're going to run around and you won't get things done. So if you are junior, I strongly advise you to have a program, a project manager whose role is solely make sure things get done.
Hi, Greg Tejan, co-founder CEO of startup Revalia Bio, also a performer under Catalyst. So I have a sort of question around the teaming structure, which I think sounds a little bit different here. If we have teams that are already working together, sort of the scheme that you just suggested, where across different institutions, there's like a prime that should submit under TA2 versus under TA1. Can you say a little bit more about the right way to propose these? Because I didn't quite follow that. Because we have teams that probably could fit in one or the other. Should we be submitting multiple proposals and talking about how we'd work together? Or can you just provide some guidance around that?
Yeah. So and I'm going to want Danielle and Gina to also kind of chime in here and correct me if I say anything wrong. So we're looking for separate proposals for TA1 and TA2. At the solution summary stage, you can submit as the prime to multiples. However, at the full proposal stage, if encouraged, say, company A submits as the prime for both TA1 and TA2. Okay, great. And then say both of those are encouraged to move forward to a full proposal. Then company A has a decision to make. Company A would have to say, okay, which one do I want to still be prime on? And maybe the other one, then they move to being a sub, and someone else from that consortium moves up to being the prime. That's how we're planning to do this. Do you guys have anything you want to add? Does that explanation kind of make sense?
I think it does. And as you were talking, I got a bit more clear. The question that I was also wondering was, as we're submitting those prime proposals, we should acknowledge the subs and the team, the other folks that we would be working with, even if they are prime on another.
Yes, definitely. Yeah. So your solution summaries would say, like on your cover page, it says, who the prime is and who are the other team members. Again, that's that cover page does not count towards that 4-page limit. I want to be super clear about that. There is a specific format of like what it looks like when you guys go to submit. It says like your title and your institution and who's the technical POC, who's the contact POC, and then who are all the other team members. And so there will be that list.
Perfect. Thanks so much.
So just to clarify, I know some of you are not exactly thrilled that we're forcing you to propose across a TA. And we went back and forth. Initially, we thought we are going to allow people to propose to sub-TAs, which would have been a more normal thing to do. But I want you to count. There's 700 of you in this meeting, right? We simply do not have the bandwidth to evaluate 700 proposals. The only solution would have been to go with AI, which we abhor. So that's why we required you to go across a TA with the understanding that we may just keep one part that we like. Okay, so that is the explanation, because otherwise we would have either been reviewing till the cows got home, or you would have gotten a review that had nothing to do with your submission.
Great. So hi, I'm Andrew Schaufstall from Case Western Reserve University. So first of all, thank you so much. This is a grand challenge and I'm really excited about it. I have a couple probably minutia questions. You've answered most of my questions in the last 2 seconds. In past proposals, there's been a component where it's required to include an ethics consultant for ARPA programs. Is that a consideration here? Is that a requirement? And would it be desirable?
Not a requirement for STOMP.
Okay, easy. And then the second one is within the first summary.
I mean, I guess I would
go.
say if you're proposing a TA1, if you're doing the TA 1.3 and you're doing human subjects research, then you obviously have to have an institutional review board, right? You'll have to have an IRB. So there's sort of your ethics component, but not separate from that. So to be very clear.
Thank you. Yeah, agreed. I was thinking of more like a broad overarching project one, and that sounds like the answer is no. The second one is on the summary proposal budget. There's rolled up numbers for TA. I'm thinking of for task area 2.1 and 2.2. Do you want just full rolled up numbers, or can we break it down into subtasks, and would that be useful?
So for the tables, I'm now I have it in my brain of what Sean presented. So basically, there's like a little mini table for each technical area and like TA 1.1, 1.2. And so that would even be that's the level of budget that we need at this point.
Just fully rolled up.
Yep, correct.
And then very last question is, there's other things that don't fully fall into like a technical area that would be more like program management that would be across technical areas. Do you want us to average the costs across, or do you want us to create a third bucket of just like general program?
Just kind of roll that in across.
Okay.
That's fine. Yeah.
All right. Thank you so much.
We'll worry about that when it comes to a full proposal in the Excel spreadsheet.
Hi, I'm Stacy from NYU. I just wanted, I had 2 quick questions. One was just to clarify the response to the previous question about the prime for TA1 and TA2. So are you suggesting that's at the institutional level? Like, just so to give you a concrete example, like if I wanted to
that's
be,
at the PI level.
It is. So if like my lab was interested in being PI on a TA2 at NYU, but then his lab at NYU wanted to do TA1, that we would both be allowed to proceed, or the whole institution can only.
I think so. Okay, they're nodding as well. So great. That's always the most important thing, is that the agreements officers concur. So yes,
Okay.
at the PI level.
At the PA. Okay, and then my other question was, I was watching back to the EPA announcement about the program, and the preceding announcement on the EPA website was about the initiative to move away from animal testing. And I was just curious how that syncs with the goals of this program, and whether there would also be more interest in projects that are not using animals.
Yeah, it's a great question. And I think, across the entire ecosystem in the government, Health and Human Services, EPA, there is a move towards getting away from animal models. We're not at that point where we can completely divorce ourselves from animal models yet, full stop. We have investments, here at ARPA-H, like Andy's, Catalyst program, that's looking at new technologies for, getting at ADME talks that are devoid of animals. So like in silico and computational models and in vitro models. That program, is still ongoing. Obviously there's been a lot of investment from DARPA over the years that's spearheaded, like organs on chips and things like that. In my mind, though, that still does not get us all the way across the finish line. And especially when we're talking about contaminants that are being ingested and inhaled, and we need to look at biodistribution and understand trafficking from that point of ingestion or inhalation towards those end systems, you kind of still need an animal model. That's kind of my viewpoint about that. But proposals can discuss if you feel that there is a technology that is able to still get at the questions that we're posing in the ISO that don't use animal models, then please defend that choice.
Hi, I'm Jason Firing from Draper in Cambridge, Mass. Asking a question about 1.2, where there's a requirement for a 15-minute assay, essentially. I just wanted to hear if you have any more guidance on the context, on the sample prep, Is that going to be deployed any other location than the performer's location? Sort of a little more explanation of the technical requirements for that sort of device or process.
So we wanted to make sure that whatever you develop for a clinical test is something that will be accessible to everybody, right? So if it takes 3 hours of physical intense work to get a result for that test, then that will not cost $50, right? The time in and of itself is not much of an issue as long as it doesn't involve human labor, right? So I would prompt you to focus on the cost, right? So if you need to let the sample on the shelf for 24 hours, but the physical, the work that somebody would have to do or the expense of the instrument that would be needed for getting the result is low, then in that sense, having the sample sit on the shelf for a day is not a problem, right? So for that purpose, I would encourage you to focus on the cost and everything will drive down from there.
Thank you. That's helpful. And so in terms of the actual performance of the project, though, it would remain at the performer's site. It doesn't have to be a deliverable instrument to other locations and anything like that.
No, but the intent is that will become a commercial.
That should be the vision, understood. Yes. But at this stage, just for understanding. Right. Yeah. Okay. Thank you.
Alexandra Rose from Arizona State University. So I wonder on quantification, both in either mass or particle count, is there some quantification limit that you have in mind that proposers would have to meet?
Well, again, it depends. I have talked to, so again, we will be paying CDC and they will be part of the project to evaluate all the methods. It's sort of, again, a catch-22. What is it that I'm testing? If it's urine, then you can start with, 50 mils and concentrate them down. It's not an issue. But if it turns out that what we measure in the urine is not correlated to what's in the tissue, then you're going to have to do tissue. And in that case, you won't have that much, right? So it's sort of, we'll learn in the process, but I would think you have to start with 1 mil of equivalent of 1 mil of whatever it is at most. Oh, how low you need to go in terms of number of particles or mass. We didn't set a metric related to that. It will be related to typical exposure.
And we also might learn more about that in TA2 as well. That's when I was talking with a lot of you guys answering questions, you guys are posing things that we struggled with a lot when we were crafting this program and trying to set metrics, because there are so many chickens and eggs and catch-22s and black holes. And it's hard to set like a lower level limit of detection if we also don't know what's the limit of harm in the body. And so they kind of go parallel. So I know that's not super helpful for you guys for proposing solutions. But yeah, I guess that gives us an insight into like how we were thinking about this problem. And I guess, proposing what you think is the limit of detection of your technology and discussing, how you would assess that.
Gabe Meister, Battelle, Columbus, Ohio. I have two questions. The first question is, you use the term acute and chronic in TA1, and that can mean different things to different folks. You have this matrix, which is very well designed. Thank you for that, without having to spoil the ocean. I do have a question, though. You have a 30-month time frame on TA2, I think, if I'm not mistaken. So I'm assuming that when you say chronic, it's going to be probably 6 months or longer, but that affects the budget. And to your point, 144 million divided by 3, divided by the performers. Just kind of curious if we should put a limitation on what we call chronic.
This is where I wish that I had my ISO in front of me, and I probably should have printed it out. So bad job, Shannon. We have a table in TA2 that does have a notional timeline of what that would look like. And yeah, so with the chronic exposure, I want to say, shoot, did we say 6 months? Adrian, you have your computer open in front of you. Yeah. Okay, so I'm going to repeat what Adrian said since he didn't have a microphone for everyone online. Yeah, so we had a timeline of chronic exposure spanning from about month 3 to month 24. But remember that also with the reference materials, it's possible we won't have everything for you guys all on day 1, that they might actually be a little bit staggered. So there might, if we're talking about, 6 different materials, there might be 2 at month 0, and then another batch at month 3, and then at month 6. So kind of bear that in mind. And then also factor in then in that overall timeline of like, what is it going to take also to analyze the results, right, of your chronic exposures. And so I totally recognize that TA2 is like super compressed because it's only 30 months as well. And so that doesn't leave, a ton of time for, especially if you're talking about chronic exposures, to then say, okay, we're going to run like a 24-month study, then analyze those samples, and then go into mechanistic understanding. So a lot of the mechanistic studies and those toxicology studies are probably going to derive a lot more from those acute studies, right, initially. But we also want to look at what happens at biodistribution and accumulation with a longer exposure timeline. That was the thinking there. Does that answer your question?
Yeah, thanks, Shannon. That's appreciated, much appreciated. But then my second question is about cost share. So I know that you noted FFRDCs are unable to be performers unless you've exhausted all their possibilities. This kind of leads into my cost share question. Are there limitations into what can be used as cost share, such as R01s, other U.S. government funded contracts, or government funded partners we work with?
Yeah, so there is no limitation on cost share. So you're able to propose either cost share in cash or resources such as personnel, IT, equipment, materials, anything that you believe would benefit the overall program in your proposal.
Even if it comes from another U.S. government agency?
In that case, it would be an interagency agreement that we would enter into if it is not a non-government entity. So for a government entity, it would be an interagency agreement that we would have to agree to.
Okay.
Sorry, are you asking about like resources that another government entity is providing already?
No, I'm talking more about resources that we may have available to us through other government contracts to do very similar work. That's why I was kind of asking the question.
Right. Okay. Yeah. So I think then the thing would be to say that, like, if there's a specific subtask that is work that's already being funded, again, yes, we don't want to duplicate effort. We're good stewards of the taxpayer dollars. We are also taxpayers. So we want to make sure we're not, doing any sort of double dipping. So yeah, if there is a specific activity that's already being funded by something else, please note that.
Yeah, and I could add to that anything that's already being funded with another agency cannot be proposed as cost share. You could just, like Shannon stated, you would just note it that this is something that's being funded via another vehicle, another contract, but it would not be considered cost share.
The one wrinkle I would maybe add to that, I suppose, is if the activities that are being funded through another source, if that is not meeting the STOMP timelines and additional resources are needed in order to meet our timelines, and be very explicit about that, about what's already being funded, and if there are additional resources needed to meet our aggressive goals, then please note that.
Hi there, Leo Trisande, NYU. I have 2 completely different threads of questions. I'm going to just try one and then you might shoot me after the first. You could imagine a living donor study being eligible for 1.3. And that begs a interesting question. Insofar, there are lots of ethical and other hurdles to think about this, but imagine if you could propose a intentional exposure study that would then be applicable to 2. I guess I want to understand whether that would be something you would consider. It's, obviously out of animals scope, but I thought, and maybe I'll just pause there and see if you want to respond to that question. I'll come back to the second point.
So, wait, we're talking of, like, you're saying living donor as in like actual human subjects research. Correct. Versus, because I know that there are other systems that are quite rare that look at individuals that are brain dead that have decided to donate their body to science. So you're not talking about that. You're talking about actual, like, living, breathing human beings, not kept alive by machines. Or are you talking about both?
Let's start with former as a simple, maybe that is simpler.
Yeah. So the former is interesting, but is tricky because we're all exposed to microplastics all the time. So the control there is difficult, right? Unless you're finding donors that, like, live in the jungle somewhere that haven't been around, like, modern technologies, right? And haven't been exposed to plastics. But even that I find it'd probably be hard to find people that have no plastics in their body. So that the control issue is tricky, and would also apply. Pardon? Well, right, but you would also want to, even if you intentionally exposed individuals, you'd want to know what their baseline is, so you know what you're applying on top of that, and that's difficult to control and understand, like what people's prior exposures and how would you measure that as well. It just the control issue, and as even aside from the ethics, just the experimental controls seem really difficult for me. Yeah, do you want to weigh in anything else?
I mean, I don't think we have said this in the ISO. So if it's not spelled out, there is no rule either way. So if, again, the TA 1.3 is open as far as you're not violating anything that's spelled out. I'm personally not too keen on exposing people.
Understood. No, I mean, you asked us to think bold, so I figured I'd ask a bold question.
I mean, there are some limits, right?
Of course, which is why I'm asking the question. No, I appreciate it. And I'm not trying to pull some science fiction madman nonsense.
Yeah, no, I mean, I totally get it. And I think even if we put the ethics aside, just for me, scientifically, the control question, how that would be addressed is something that still feels kind of like a non-starter. If there's a really strong justification for how that would be addressed and how the ethics of that would also be addressed, that, I mean, we would review that certainly, but I'm skeptical of that. But we'll read all proposals. They don't go to AI, they go to us. We read them.
So I think my second thread of questions, if you'll permit me, after having thrown that, is a bit simpler, which is I want to understand for TA2, would there be space to explore the measurement of monomers in addition to micro and nanoplastics as defined? And would that then, and I guess in general, that's just, I think there's a scientific question to think about there. In theory, in the human body, we could get I don't know if we could get all the way down, but something that I would just think about exploring from a measurement perspective. I don't know if that's in scope.
Defer to our resident chemist.
Yeah, I think it's a good thought with monomers. The materials that we'll provide from the references will not have any monomeric units on it. It's something that we're going to control for. I think, one of the things that we mentioned earlier is that we aren't limiting you to only test the materials that we provided. So if you do see an opportunity that monomers could be an interesting avenue, we would be open to you testing that as an additional material. But yeah.
I'm Enid Neptune from Hopkins. I have a couple of less ethically problematic questions. So the first builds on kind of the query about the chronic exposure kind of systems, because the question is kind of the phase of clearance and what the time frame that these models should be monitored in terms of clearance after presumed accumulation. And that type of protocol structure would obviously be far more kind of prolonged than just 6 months. So I wondered if you had thought about that and have suggestions. That's question one. I don't know whether you want to address that issue.
Yeah, let's maybe just start there, because that was already like a lengthy question. So let's do that while it's like on my brain. No, I think that's a good question, and we don't have any specific requirements about that. So if a proposal, would describe then what the exposure looks like, what are the time frames at which that system, whether it's in vivo or in vitro, is monitored, like you said, both for clearance as well as, trafficking, right? Because that has a time component as well. So that experimental design should be very clearly spelled out and justified as to, again, what the exposure actual looks like, what the follow-on period looks like, what the analyses look like, what are the samples, and how are you going to be addressing things over the time course. So we don't have specific requirements, but just propose what you think is the best system to look at that.
Thank you. The second question is more about this whole issue of multi-organ surveillance in terms of trafficking, because kind of it's very difficult to predict what you might see with serial imaging in terms of where the primary accumulation will be. And so if you're interested in having resident expertise, at least within your team structure, is that something that's going to be iterative, that you can say, these experts are available, and if we need them, we'll bring them in so that they can look at injury or toxicity measures? Or how should that be done and incorporated in a way that kind of gives us the best guidance?
Yeah, that's a really good question. So we did half address that, but not fully in the ISO. So we did discuss how, early in TA1, if you're looking at, whole body imaging, because again, we don't know what we don't know, right? There have been studies that, have found micro nanoplastics in kind of almost everywhere that we've looked, but we haven't looked truly in a matrix style fashion across polymer types and sizes to really figure out exactly what goes where. So we don't know what we're going to find. And so I do recognize that it might make things a little bit difficult then for a full proposal if you don't know what's going to end up where and what's going to be causing the most harm for follow-on mechanistic studies. Yeah, how do you know who to put on your team? And so I think, a way around that might be kind of proposing like your best hypothesis as to what you might find and sort of budget in that way. If there are tasks that would be, like, you could imagine a similar set of tasks and activities, though, that might still be common regardless of organ system, right? So that you would have maybe tasks like if you find out that it ends up in like lung and liver and that's where you want to focus, what would those tasks be and what would that budget look like? And then if there's interchangeable personnel, or you say like, this one expert might be pulled in, but these same tasks would occur regardless of organ system, like what I could think about proposing something like that. Does that make sense?
And also bear in mind that you can change during the period of performance, right? So you can budget, hey, I'm going to get a nephrologist and a GI physician, but if it turns out the brain is more of the problem, then during the period of performance, you can swap.
Yes. Thank you, actually, Alana, so much for making that point. So again, I mentioned earlier, I've been in the ARPA sphere a very long time. It's very rare that we fully fund like a proposal exactly as written. It's also very, very rare that a contract never gets modified. This is a 5-year program. Sean has worked with me for a long time at DARPA before. We managed one effort where I think it got modified 27 times. Now, this was also COVID, and this is a pandemic preparedness like type of program, so it got modified a lot, but that happens quite frequently. So, propose your best, guess as to like what the statement of work and associated budget and personnel would look like, with the understanding that things happen. Science is not linear. Things can fail. Things can work like gangbusters, and we want to divert more resources towards that thing. Like, we modify contracts and agreements all the time. So you're not locked in from day one.
Hi, Aaron Essercon, University of Chicago. I had a question about sort of the TA2 and TA1 and the ambiguity around sort of plastic-free usage in the studies. So you're asking for a lot of things where you would modify the particles or chemically alter them or track them in some way that's radioactive. Once you've altered them, do you still need to maintain polymer-free workflows all the way through? Because presumably then you would be able to distinguish between what was labeled and what was unlabeled, but it's not clear from the ISO, unless I missed it somewhere, and I apologize, it's a very long document, that's the case. So I just wanted to get some clarity on that.
Yeah, that's why I tried to use the terminology both minimize and mitigate. So what you're talking about, if you're having labeling, then that is kind of a mitigation strategy, right? Because you can more clearly identify what you have put into the system, especially at least for like biodistribution type strategies. Now, if we're talking like toxicity, you would want to have a much clearer standpoint of like minimizing extraneous contaminations. That's why I use both of those terms, because I think they're both really important. Minimizing as much as possible and recognizing that nothing in science is like 100%. There's always uncertainty there. And so how are we going to mitigate as well?
Thank you for having us. I'm John McNeil. I'm a cardiac anesthesiologist at University of Virginia. Don't have a question so much as a comment that addresses some of the other questions. It's not really ethical to expose people to plastic, we don't think, but I will point out that in modern healthcare, we do a lot of iatrogenic healthcare plastic exposure. So in cardiac, for example, the heart-lung machine's all plastic, the plastic reservoir is plastic. And so you have control groups of some patients that have surgery and some don't. Or if you get a heart cath, we think your microplastic serum levels go up by 20 times. Some patients get cath, some don't. So you have kind of natural controls in the hospital. And then for degradation and excretion studies, you have the half-life studies. These patients oftentimes have arterial lines, you can draw blood very easily. So I'd encourage you to look at healthcare and the hospital for these studies of looking at how do we control the exposed plastics versus not, and also how do we measure the excretion in these patients.
Hi, Aaron Stubbins, Northeastern University. I know you're going to provide plastic standards, but false positives are a big problem. And so for trying to getting samples of biological tissue with guaranteed no plastics would be a great asset to the group. I don't know if that's something, we can prepare that individually, but lipids, other things like that are going to show up by many of the methods as plastic. So is there thinking around that?
That's a good point. I'm not exactly sure how you're going to breed mice that have zero plastic exposure. The idea was that you will have two groups. One will be control and one will be purposeful exposure, right? The control may not be completely controlled, but at least you have a baseline that's, you know that what you inflicted on them is above and beyond that. If you in your research design, if you can think of a way to design a plastic-free mouse, I think it's worth considering. In the end, again, I'm relying on that discussion at the very beginning of the program where we're going to solidify this a little more.
Yeah. And the other part of your question, though, about lipids are going to maybe look like plastics. That's the whole point of TA 1.1 is to develop those gold standard measurement techniques such that you don't have as much of the cross signaling.
Yeah, my question was more related to TA 1, where how do any of us know that our sample is plastic free? So we go to a sample and we measure plastics in it. Pyrolysis, GCMS, other methods can measure very precisely, but the main problem with them is false positives. And it's false positives of basically lipid-like structures. And that's an issue.
Again, we divide it, so we assume that your baseline will be whatever it is, right? You can think about spiking your samples, right? So it's the delta that we look at more so because it's more accurate. And if you spike your actual samples with C13, for example, that you know that what you put in, so it's the delta that matters.
Yeah, but then when we move to real samples in the clinical or other settings in 1.3, any of those methods trying to measure plastics, the baseline is the real person, and we still don't know what the false positive is in the real person. That's kind of the central challenge to measuring plastics in people is the false positive. And I was wondering if there was a...
Right, and that's why you have the gold standard team under TA 1.1, right? And they will have realistically 6 to 9 months, by the time you have your IRB and you start enrolling, it will be month 9. And by that time, hopefully that gold standard would have been established. May not be gold, maybe, maybe silver, but it's better than brass, which is what we have now, right? So in that sense, that will be as good as we have at that point in time, and we can consider having that team analyze the samples for everybody.
Yeah, if someone can make some plastic-free lipids or something, that would be an awesome addition to any reference material.
All right, I think we have time for maybe 2 more questions. I know we've already gone way over time. This discussion has been phenomenal. We're going to close Q&A at 12, and then we'll break for lunch, and then we'll come back for the TA3 session, but we can take a couple more questions.
Well, great. Let me make one statement now because I will forget. Okay, so when you write, so for the abstracts, it's less important, right? You have 4 pages, you're trying to cram in as much as you can, it's fair game. When you come at the proposal stage, I know there are many companies here and you have concerns about what you write there being leaked out. And obviously you don't want your technology being leaked out. I want to assure you that review is 100% confidential and the proposals and the abstracts are not subject to FOIA, right? We I'm a great believer in what Ronald Reagan was saying, trust but verify. We do not trust, okay? So if you're saying you have a method that does X, unless I see the data, the default is I don't believe you, right? So if you want your proposals to be funded, you need to explain to us exactly how it's done, and you need to show us the data. Don't tell me, believe me, I have that. We had a few wonderful proposals in my last program. I honestly believe they had that, but we do not fund black boxes. So if you're telling us I have this amazing method that solves your problem, I don't believe you. If you give me the method and the data, then yes. So please, all of you bear in mind, the proposals are confidential. The data is confidential. It's only government employees seeing that. We're under an NDA, and those are not subject to FOIA.
One other thing, I guess I'll just say kind of along those lines too, again, doesn't matter as much at the abstract stage because you are so space limited, and we totally recognize that. At the full proposal stage, don't bury key supporting data in the references. If there's something that's, kind of tangential to your work and supporting, then you can obviously put that in the references and we can look at that. But if it is like something that is seminal to the work that you're proposing, don't bury it in the references, put the figure in the proposal and put the data in the proposal so we can actually see it. That's really important.
References are not subject to the 4-page limit. So you can put as many as that, but again, it will depend how many abstracts we get. If we get 500, I can assure you we will not read the references.
Human, guys.
Great. I guess I have the luxury of one of the last questions. I'm Christine McDonald from the Cleveland Clinic. I have some questions about TA 1.3 in terms of the samples. Are these expected to come from all healthy individuals, or how are we going to manage the comorbidities? Because that may change the uptake and distribution of microplastics. If you have inflammatory bowel disease, your gut might be leakier. If you have heart disease, you'll have other issues that way. Is that going to be an important consideration in those studies?
It's up to you how you design your study. The main goal of that study is to look at the correlation between blood, urine, and tissue, right? So in that sense, we actually want a range. So if you can design your study such as due to occupation or pathology or something else, you suspect that a particular subcategory has more, then we definitely want that included, because at the end of the day, we want to be informed whether we can sample blood or not.
Thank you.
So I'm Prabir Roy Chaudhary. I'm a nephrologist at the University of North Carolina. I'm still sort of struggling with 1.3 in terms of the tissue that is best to be collected, because on the one hand, clearly I'm hearing that it would be great if it's a tissue that could be sampled from a large number of people, which really means that you're going to have a biopsy from a healthy person or a relatively healthy person who may be at risk of micronanoplastic accumulation, which really limits you then to colon or skin. And on the other hand, and I just want to pick up on the comment made by the cardiac anesthesiologist, there are so many settings in clinical medicine where there is huge exposure to microplastics. So ECMO is one, but another one that is my life are $600,000 as patients who have 100 liters of their blood exposed to plastic tubing 3 times a week. And there are so many innovative ways that one could leverage that to actually get a better idea, at least on the clinical side, about the correlations between blood and tissue. The problem is that these are all niche populations. So
Yeah.
how do you reconcile those two things?
Yeah. So again, the question that we're trying to answer in TA 1.3 is kind of trying to help TA 1.2. So TA 1.2, again, our goal, what we conceived in our brains was we need to have a fast and cheap and readily accessible test, right? And we were assuming that was going to be blood or urine. And then we were like, wait, what if blood and urine doesn't actually correlate with tissues? And so TA 1.3 has to address that. And so we're not specifying what tissues you want to look at. If there are ways of looking at, like we brainstorm, we're like, okay, like what are tissues that could be accessible for a clinical study? That could be like an appendectomy, right? That could be like placenta after birth. That could be elective cosmetic surgery and there's tissue left like that. Like those are ways that you can get at the intellectual question of blood versus tissue. But then that still doesn't solve the problem if there isn't that correlation between blood and tissue or across different types of tissues. Then what do we do for our like actual clinical diagnostic? And that's what kind of then led us potentially towards that colonoscopy element. But we're very open to, what you guys propose for TA 1.3. And the important part is there a correlation between blood and tissue, and answering that question. So we're open to a lot of different approaches as to how you might assess that.
Again, so the point is to ask a question is to be able to act on the answer, right? So learning something for the sake of learning is great, but if I can't do anything about it, then why learn about it, right? It's not helping me. So for example, one obvious solution is to do a cadaver study where you can sample multiple tissues, right? And you may actually learn in that study that, well, maybe colon is correlated with blood, but maybe brain is not. And I think to me, that's a very useful study. And I can see how that would be part of what we fund. But in the end, at least a portion of that study has to be actionable. Otherwise, science for the sake of science doesn't take you far.
I think we're at time. I do apologize. I know I still see a few hands up. Reminder, please submit any other questions to the STOMP inbox and to the FAQ portal. We will address them there. Again, for folks online, again, please do the same. Make use of the FAQ portal, and we, again, update those, multiple times a week. We're going to be posting answers, from today's Q&A session on that FAQ portal as well, so stay tuned for that.
So one more thought. I wanted to reiterate when we tell you discourage means discourage. And I want to tell you that I've been a program manager for 3 years, and I personally have never ended up funding somebody who I had discouraged. You may think that you have a great submission. The data that you do not have is what else was out there, right? And you can imagine with the number of people we have, honestly, I have some proposals for my prior program. We feel bad about telling them no. Honestly, they're that good, but we don't have infinite resources, right? So to us, when we tell you discourage, we encourage significantly more, a multiplier of the number of teams we fund, so we have plenty to choose from. But when we tell you discourage, please consider it. 4 weeks of your time is a lot of time to put night and day into writing that if truly you don't have a chance. Thank you. So we are going to start the brainstorming session. So for those of you online, I think this is the end of what you can participate in. The brainstorming will go for about 40 minutes, and then we'll have the poster session for which you cannot participate. Thank you.